Okay, good morning, everybody. We're going to pick up where we left off in chapter 19 with the bronchial tree, and this particular voiceover will go through the end of expiration there on page 747. So we're starting on 738. We talked in the last voiceover about the larynx, the presence of thyroid, cricoid, and epiglottic cartilage. We also talked about the 20 or so C-shaped rings of the trachea. And so as we look here in the diagram, we're encountering what's called the bronchial tree. This is sort of like an inverted tree. If you use your imagination, the trachea would be the main trunk of the tree. And then as it begins to split into the left and right primary bronchi, those are like giant branches coming off the main trunk. And of course, we see continued subdivisions of those branches into smaller branches and so on and so forth. So that's where it gets the name bronchial tree. Where the trachea splits is referred to as the carina, and it splits into two primary bronchi. That's the plural term, bronchi. If we're talking about one, we use the term bronchus. So obviously here's the left primary bronchus, here's the right primary bronchus. Those bronchi in turn split into several secondary or lobar bronchi. And notice on the right side of the lung, which has 3 lobes, we have 3 secondary or lobar branches off the right primary bronchus. And on the left side, with just 2 lobes, we have 2 lobar or secondary bronchi. And those secondary bronchi in turn split into what are called segmental or tertiary bronchi. You can look on page 739 of your text, and it lists there those branches of the bronchial tree. From the tertiary or segmental bronchi, we have what are referred to as terminal bronchi. And from the terminal, we branch into what are referred to as the bronchioles. And you'll see a diagram of that coming up on the next slide or so. But again, just note again the 3 lobar or secondary bronchi on the right side and the 2 lobar or secondary bronchi there on the left. This is just reiterating what we just spent a moment talking about in the previous slide. I've outlined in number 1, 2, 3, and 4 those ever decreasing sized structures branching off of the trachea. Note again 2 lobes on the left and 3 lobes on the right. As we delve down into the microscopic structures of the bronchial tree, remember the terminal bronchi, or bronchioles, excuse me, bronchioles now, not bronchi, bronchioles. We use the term bronchioles. Terminal bronchioles will in turn branch and split into respiratory bronchioles. We can see one respiratory bronchiole here. Note about 1/2 millimeter in diameter, so you can actually see this with the naked eye. You know what a millimeter is, right? We've talked about that, the smallest unit of the metric ruler. So this is 1/2 of one of those millimeters. So you can see it, but it's very tiny. And then it in turn branches off into what are called alveolar ducts. So there's 3 of them shown here. And then at the tip of the duct, it ends in this kind of cluster of little air sacs. And those are called the alveoli. That's the plural term. If we're talking about one, we use the term alveolus. So this is one little microscopic air sac. And think back to when we talked about this in the histology lab. We talked about the simple squamous. I showed you guys a section of lung tissue on the monitor in chem physics lab, remember? And so that's what we cut through. It was a slice through a cluster of these alveoli, and we could see the very thin walls of the alveoli composed of simple squamous. Why simple? Why flattened? One layer. Well, hopefully you remember the magic word starts with a D. Diffusion, right? It's the diffusion of the oxygen and the CO2 across the alveolus into the bloodstream. We'll talk about those capillaries in just a moment. Here's an interesting plastic cast of the bronchial tree. Kind of pretty. Note again the trachea, the carina, the left and right primary bronchi, and so on and so forth, all the way to the very tips of those branches. Right? We don't see the alveoli here. Remember, they're microscopic, but they're going to be at the very, 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 very tips of these little branches. You can't see them, but they're very, very tiny, and you have lots of those alveoli. So here we are back at the alveolar ducts here, branching, remember, into the little microscopic air sacs, the alveoli. I want you to pay attention to the fact that the surface of these alveoli is covered by blood vessels. So we've got incoming deoxygenated blood, or oxygen poor blood, it's a better term to use, shown here in blue. We'll talk more about the pulmonary artery and pulmonary vein when we get to the cardiovascular system, but basically this is blood that is very low in oxygen, so we color it blue. And when that oxygen poor blood comes to the lungs and comes to the capillaries there on the surface of all these little microscopic air sacs, the alveoli, that's where that gas exchange, where that diffusion occurs. Remember, the CO2 will diffuse out of the blood, go into the alveolus. What will come aboard the blood? Well, it's oxygen, right? So when the oxygen comes aboard the blood, it becomes oxygenated. And we denote the presence of oxygenated blood by coloring this blood vessel red. So it's called a pulmonary venule. And this is going to ultimately be taken to the heart and then pumped to the rest of the body, that oxygenated blood. So it's across those alveolar walls and into the capillary that diffusion process takes place. Okay, so note again, as the box states, there are hundreds of millions of these microscopic air sacs at the very tips of our bronchial tree. So there's a lot of surface area for a lot of diffusion. Here's a histologic section of some alveoli. We talked about this a few moments ago. So this is where the CO2 and the oxygen is going to be in the center. And then that simple squamous that lines the alveolar wall, that's going to be right here. Sorry. Anyway, it's the very outer edge of that cutaway view. I can't go back, unfortunately, here in the voiceover. So here's another diagram of a single alveolus that has been sliced. We can see the simple squamous cells. There's one here, here's one here, here's one here. This makes up the wall of the alveolus, right? These are all simple squamous. It's also shown here as well with the nucleus inside. And here's our oxygen poor blood that we talked about a few moments ago that's coming in. And remember that gas exchange is taking place between the wall of the capillary and the wall of the alveolus across that simple squamous. The CO2, as we said, diffusing out of the blood into the alveolus, diffusing from a higher concentration to a lower concentration, while the oxygen diffuses from the alveolus into the blood, right? I should have used different colors there, shouldn't I? Blue should have been over here, red should have been over here. Anyway, you get the idea that blood gets oxygenated as evidenced by the color and then taken again back to the heart. So diffusion, very important process. Movement of gases, in this case, from higher to lower concentrations. Now we're going to use a different term in a few moments when we talk more about gas exchange. We're going to use the term partial pressure instead of concentration. So when we talk about gases, we use a special term, and that is partial pressure. So more on that in a few minutes. Let's see if this video plays while we do the voiceover. I guess it's not going to here. Anyway, you go back into the regular PowerPoint slide here. I forget what number it is, and you can play this video. This is going to simply talk about that process of gas exchange. Okay, so check it out. Watch that video. So we're looking now, of course, at the ribs, the sternum, the fact that this is protecting, isn't it, our lungs. This is a transverse section through the thoracic cavity. This is the very same diagram that you had in, I think, exercise 2 of the lab book. If you think back to our second lab, we talked about the two membranes here, the visceral and parietal membranes. Now, because we're talking specifically about the lung, we got to make sure that we use the term pleura. Pleura refers to lungs. What's the difference between the visceral and the parietal pleura? Well, we said that a visceral membrane covers the organ itself, while the parietal membrane lines the cavity within which the organ resides. And between the two is a space, a theoretical space, shown here in blue. So again, here's our visceral pleura lining the lungs themselves. And then we've got our parietal pleura that's lining the cavity. Okay, the cavity is shown here in blue. The space here is the cavity. And what's interesting to note here is if you take and trace, for example, the visceral pleura, let's just trace it around, look what it becomes. It becomes the parietal pleura. See how that membrane folds back upon itself? Right, that's kind of interesting. Becomes the visceral. And as we said, between the two membranes is this pleural cavity. And within the pleural cavity is a fluid, a serous fluid, that's secreted actually by these membranes to keep this a friction-free environment. Because when you inhale and exhale, as your lungs expand during inspiration and then sort of decrease, deflate during expiration, they're rubbing against the cavity. Or I should say they're rubbing against the parietal pleura, okay, as the lungs expand and deflate. So there has to be no friction here. And there isn't because of the presence of this serous fluid. So let's talk a little bit about breathing. We do it all the time. It's an unconscious process when we sleep, when we're awake, we're inhaling and exhaling. So before we talk about the process of inspiration, let's consider the situation if we're sort of like in between an inspiration and between an expiration. So we're not taking air in, we're not taking air out, we're kind of in this in-between zone, if you think about it. You're not taking air in, not taking air out. If we look at that state, and we'll define it as the lungs are at rest. They're not inspiring air, they're not expiring air. And we examine what's referred to as the intra-alveolar pressure. Okay, now what does that mean? Intra. Intra means within. So if we could monitor, stick a probe inside the alveolus and measure the pressure when the lungs are at rest, we would find that pressure to be very close to about 760 millimeters of mercury. That unit, millimeters of mercury, is a pressure. And we're going to compare that intra-alveolar pressure of 760 at rest to the outside pressure in the air, which happens to also be, as you'll note, the same number, 760. Now this is based upon what's referred to as standard atmospheric pressure. And without getting into a lot of complex physics here, depending upon your elevation, this number can change. But 760 is considered standard atmospheric pressure at sea level. Because we live a little bit above sea level, in your room where you're sitting right now, that number could very well be higher than 760 or not the same number. Anyway, when your lungs are at rest, however, that pressure on the outside of your body is the same pressure as the intra-alveolar pressure inside your lungs. Now again, that's at rest. You're not taking air in, you're not taking air out. When you go to inspire air, what happens? Well, air comes in, right? You all know that. But let's think more about some of the nitty-gritty of what causes air to move into your lungs. We don't think about this a whole lot. Okay, so here's the scenario. You've got the lungs here, protected by the ribs. Remember, we also know that the diaphragm lies here below the thoracic cavity. It separates the thoracic from the abdominal pelvic cavity. You guys all know that. So here's the diaphragm. And there are nerves, referred to as phrenic nerves, that will stimulate the diaphragm to contract. Now, normally, at rest, the diaphragm has this somewhat shallow arching shape to it. But when the phrenic nerves stimulate the smooth muscle of the diaphragm, as the arrows show, that diaphragm is going to move downward. Now let's look at a metaphor or an analogy here in the upper right hand corner. We have a syringe. You all are familiar with syringes pretty much. And the plunger here, if we were to pull down on the plunger, we would create a vacuum, wouldn't we? And of course, as the arrow shows here at the top, air would move in from the outside. Well, in the same way that the plunger is moving down, bringing air into the syringe, when your diaphragm moves down, when it contracts in response to the phrenic nerve stimulating it, guess what? Air moves in. Yeah. So we talk about this being an active process, meaning in order to inspire air, we have to have nervous stimulation inducing contraction of muscles. In this case, the phrenic nerves stimulating the diaphragm, and that induces the movement of air in. Okay, so active means some sort of stimulation by a nerve or a set of nerves, typically to a muscle. Now, when that happens, look what occurs to the intra-alveolar pressure. Remember, at rest, what was the number? It was the same as the outside, right? 760. But when you stimulate that diaphragm, you cause the intra-alveolar pressure to drop. In this case, just a couple millimeters, but it doesn't sound like much, but that drop in intra-alveolar pressure is enough to cause air to move in, because air will always move from a higher pressure to a lower pressure. So you're creating basically an intra-alveolar pressure that is below that which exists on the outside of the body. And that's what brings air. Now, if you need to take additional inspiration, okay, so ordinarily the stimulation of the diaphragm is enough to induce inspiration 99.9% of our day. We bring air in via stimulation of the diaphragm by the phrenic nerves. But let's say we want to take a bigger inspiration beyond the normal inspiration. In that case, as the slide indicates, we would have to stimulate additional muscles. Now, we haven't talked about these yet, but we will very soon in lab when we get to the muscular system. But we've outlined these. The external intercostals are muscles that lie between the ribs. Inter means between, costal refers to rib. These are external, it means they're on the outside. There's also internal intercostals, which we'll talk about a little bit later, but these are the external. They're more external than the internal are, so they're on the surface here more so. And so when those contract, as it indicates the ribs are pulled up and out, you're expanding your chest cavity, your thoracic cavity, and keep in mind that even though we have the serous fluid keeping a friction-free environment between those visceral and parietal membranes, those membranes are still in contact with one another. Just like if you took two glass slides and you put a drop of water between them, you know, it'd be hard to pull them apart. Well, in the same way as the parietal pleura, which remember is lining the cavity, if it's moving up and out, which it will do in response to the stimulation of these external intercostals, when the ribs move up and out, the parietal pleura is going to move up and out, and the visceral pleura, because it's connected to it, going to be attached right to it, it's going to move up and out as well. So you can bring more air in as a result. Now, let's say you want to take the biggest possible inspiration you can. When you were a kid, you went to the pool, you probably played this game where somebody throws a coin in the deep end of the pool and you have to dive down, and who the first one that gets the coin keeps it or whatever. You know, we've all played those games. So if you want to take a super big inspiration, then you'd have to draw upon additional muscles beyond just the external intercostal muscles. And so this slide is showing a couple additional muscles that we'll be talking more about coming up again in another couple weeks. The sternocleidomastoid muscle, that's a mouthful, isn't it? There's two of them. There's one on either side of your neck, basically. So this is looking at the left sternocleidomastoid. There's one on the other side, too. And the pectoralis minor muscles, you'll be learning about those soon in lab. Here's their location. They're just underneath the pectoralis major, which is the primary upper chest muscle of the body. When those muscles contract in response to nervous input, nervous stimulation, look what happens. The thoracic cage, the rib cage, is drawn up and out even more. So what are you doing to the intraalveolar pressure when you do that? Well, you're dropping it below 758, considerably below that, right? And the effect is to bring in the biggest possible inspiration that you can. So here's our major steps of inspiration that we just spent some time talking about. Check those out. Those should make sense to you by now. Let's then finish up the voiceover by talking about what occurs during expiration or exhalation. Unlike inspiration, which we said was active, meaning nervous stimulation caused muscles to contract that helped lower the intraalveolar pressures to allow air to move in. In the case of normal quiet breathing like you're doing right now, when air exits your lungs, it occurs as a result of passive processes. In other words, there's not typically any sort of stimulation of muscles during normal, quiet breathing. So in this case, we're looking at a phenomenon referred to as elastic recoil. So when you inhaled a few moments ago, there was elastic tissue in the lungs and in the abdominal organs that became stretched as a result of that inspiration. When you're no longer stimulating muscles and you're no longer then stretching those fibers, like a rubber band that's been stretched, when you leave that go, it bounces back to the pre-stretched state. Well, it's the same sort of an idea here. So during the time at which the diaphragm is relaxing, So remember, it was dome-shaped, or it was flattened, I should say. It's being pulled down. When it returns back to that dome-shaped morphology, coupled with this elastic recoil, is enough for air to exit the lungs. So it's really, it's twofold. It's relaxation of the diaphragm, but more importantly, it's recoiling of some of those tissues that got stretched during inspiration that leads to air leaving the lungs. And I want to draw your attention to this notion that expiration during quiet, resting breathing, okay, as you're sitting watching TV, reading a book, driving to school, 99.9% of our lives we're doing this, occurs as a result of elastic recoil, not due to contraction of muscles. But once in a while, we may want to go beyond a normal exhalation, like if you're playing the trumpet or you're blowing up some balloons for a party or whatever. If you want to put more air out of the lungs, draw more air out beyond the normal exhalation, then guess what? We have to turn to an active process now, meaning we've got to stimulate muscles to help push more air out. And so that's what this slide kind of shows. If you want to maximally expire air from your lungs, you will need to stimulate some muscles, and those are shown here in green. Now note, we're tapping into internal intercostals here, not external, but internal, and some of the walls, muscles of the abdomen. We'll be looking at those and learning about those coming up again in another couple of weeks. The internal, external obliques, they're called, the transversus abdominis and the rectus abdominis. These are four very thin layered muscles making up the abdominal wall. When they contract, in concert with the internal intercostals contracting, that acts to squeeze the abdominal organs and more air is expired from the lungs. Okay, we're going to wrap up talking a little bit about another substance produced. Now don't confuse this with the serous fluid between the visceral and parietal membranes. We're talking here about a fluid that is produced inside the alveolus. It's called surfactant. And this little insert sketch shows an alveolus, and here is what's referred to as a type 2 or surfactant secreting cell. This is not one over here. Okay, this is something different. But we'll talk about this guy right here. And this particular cell produces this chemical called surfactant, which decreases the surface tension of water. Meaning that when you inhale and expand those alveoli, you do so without any problem because of the presence of this chemical. This surfactant allows the alveoli to reinflate effortlessly during inspiration. However, if a baby, a preemie, is born, it often has trouble breathing because the lungs are among the last organ systems to fully develop in the fetus. So if you're 4, 6, 8, 10 weeks premature, there can be lots of health issues, not the least of which is the baby is unable to breathe on its own or has a very difficult time doing it. And so sometimes it has to be put into a special cubicle within which this chemical is given into the airspace that the baby's in, and the baby will inhale this surfactant because it can't produce it. Its type 2 cells aren't mature enough yet. And so that's going to allow those alveoli in its tiny little developing lungs to reinflate more easily. So prior to medical marvels, many a premature child has died because it just couldn't breathe normally. Okay, this I think is the last slide of this voiceover. It's simply reviewing the various steps of expiration, so check that out. And I think that concludes this particular voiceover.